Hey, so thanks everyone for coming out on this cold but beautiful day. So normally whenever I give a talk in kind of a formal setting like this with a podium and up on a stage, I can feel a little bit anonymous, but looking around the room I recognize a scarily large percentage of the people here, so I feel a little more nervous than normal. So if I wander too far off track, don't hesitate to just stop me and ask a question. So it's a pretty informal talk today. And what was exciting about having a chance to speak on this topic is that almost always whenever I give a research or extension talk, the growers and the farms that cooperate with our research get collapsed down into one more N in the sample size, essentially. We don't really have a chance oftentimes to really highlight the fact that none of the work that we do right now will be possible without the collaboration of our growers that work with us. So there's essentially no research in my lab right now as of today that isn't being conducted on the farms of cooperating researchers, farmers, at least in part. So with that, I wanted to start with just talking about some of the more bigger questions, kind of pan back to what we're interested in my lab. So this is sort of a typical agricultural monoculture where you have just very, very low biodiversity, the single crop plant, and the nearest more natural habitat, if you can call it that, is this weedy field margin kind of way off on the horizon here. So these, of course, are quite simplified habitats. And from some of the earliest days in agroecology, people have thought that maybe it's something about this extreme simplicity of these habitats that leads to the common pest outbreaks and other problems we see in big monocultures. So, for example, if you go to a tropical rainforest or some other highly diverse natural system, you're almost never going to see all the plants stripped of their foliage. And, of course, in a lot of agriculture, particularly big monoculture, conventional agriculture, that would happen almost every year if we didn't use insecticides or other interventions. So it might be something about this ecological simplicity that leads to some of the problems that we see in big modern farming systems. So just to take one example, so my lab works a lot with biological control and natural pest suppression, and we know that a lot of these natural enemies can benefit from being in very diverse systems. So for example, a lot of predators and parasitoids, so important in pest control, will also feed on pollen or nectar if those resources are available. They also are going to need prey outside of the growing season to maintain themselves and habitat and shelter to overwinter and so on. So natural enemies and biocontrol is thought to generally be stronger in a more diverse system for the obvious reason that it's just easier for the natural enemies to survive under those sorts of conditions. And I think you can take it back to sort of a broader point. So within so much of organic agriculture, kind of one of the underlying principles is that biodiversity is good. And explicitly, this is often part of the standards that establish organic certification, is that you need to try to have more biodiversity on your farm for biocontrol agents, pollinators, and other species. But what my lab tries to do is to try to figure out what are some practical ways that growers might be able to manipulate their farming systems to add in sort of just the beneficial types of biodiversity. So we know that the other aspects of biodiversity are going to be things like plant feeding nematodes and weeds, things that we might not necessarily want. So how do we attract and encourage the good biodiversity and not just bring in bad biodiversity? So it's a kind of a complex ecological question to try to get at that. So kind of the setting of this whole session this morning is, like so many university researchers, as Colette just mentioned, there's a temptation to do a lot of your experimental work on the university research farm. So things like here are some of our cage experiments. These are big meter-cubed cages. So these are big field cages where we can dump in whatever plants and insects we want and wait to see what happens. In this circumstance, we control everything that's happening. We control what species are there and can kind of gather data relatively quickly that lets us get papers and get tenure and not get fired and stuff like that. So that's important, of course, but it also, what's importantly, is that it allows us to understand a lot about the mechanisms of how the different insects are interacting in a really detailed way. But the obvious concern that we always have is that, does anything that we're doing in these cages represent what happens on a real farm, where you don't control any of the species that are there, as growers well know, and there's a lot more complexity than what can be in a cage. So in the last few years especially, we've tried to do, as I mentioned, as much of our work as possible, and really now we're doing all of our work on the farms of cooperating growers. So there the conditions, of course, are perfectly realistic. They're real, actual working farms. But the challenge is to generate data that are interpretable so we can learn some general principles that we need to get to within ecology. Okay, so Amanda, my wife, is the person who invited me to give this talk. So I was going to say, this is unjustified nepotism at an almost Trump-like scale that I'm up here giving this talk. But she helped to lay out an outline that I thought was helpful to kind of guide me through the talk. So I'm going to stick to exactly the discussion points that she asked. So this is the first one. How did you get involved in on-farm research? All these pictures, I believe, are at Diane Green's farm. I know Diane is here somewhere, right there. And really the truth of almost everything that happens in my lab is, I rarely, if ever, actually get out onto a farm nowadays. This is actually me on a farm. Whoops. Oh, no. Me on a farm right here, but that's a rare sight. The people that actually do all the work are the students and postdocs in my lab. And this is Renee Prasad, probably not the most flattering picture she would want to have included here. But she was the very first PhD student in my lab, and she was interested in a really conceptually simple type of predator conservation approach, which is beetle banks. So beetle banks are trying to address the problem in a big monoculture. You don't have any natural habitat for the natural enemies. And so beetle banks are these grassy strips that you run right down the middle of the field. And so that's a place where ground beetles and ground crawling spiders can overwinter and find food and find refuge from tillage and so on. So she was interested in that predator conservation technique, and she was from Vancouver, BC, and already had been working with farmers as a crop consultant. And so she wanted to do on-farm research, and so that's what she undertook to do. She put in a series of beetle banks on about a half dozen farms, so she had paired fields where one field had a beetle bank and one didn't, and looked to see how the predators' numbers would change and whether that had any impact on the pest. And here's just a little bit about the pests. So she was interested in root maggot flies. This might be unfamiliar if any of the growers, or particularly from the west side, might know these pests. So they're little flies. The maggots drill into the below ground parts of the plant and can do damage. So Renee had heard from her grower. So here's a radish that was not attacked by root maggots. Here's a radish that was attacked. And her growers were telling her that this radish is more valuable than this one. Okay, so what's good about these is there are some predators on the ground that can actually control these pests. And in particular, ground beetles or carabid beetles. Really important predators on farms, but they're often nocturnal, and they live in the ground a good bit of the time, so they're often missed, but could have a strong impact. And this is just the predator that these beetle banks are supposed to benefit. And so again, these are on-farm data. This is not a research farm data set. So here she's tracking beetle densities throughout a growing season. This is the number of these predatory beetles. These are our good bugs. And here's a field that had a beetle. Here's the average across the fields that had beetle banks, and here's the ones that didn't. So she was finding out in the crop dramatically higher densities of ground beetles. So it seemed like the predator conservation was actually working. I'm not going to show the data, but I'll just summarize it. What was troubling is when she put out fly eggs to see if more flies were being eaten, there was no difference between the two treatments. So she had dramatically higher numbers of predators, which normally is where research stops, but the actual pest control wasn't being improved at all. And through a sort of agonizing series of observational studies, she found that this was the problem. So this is an invasive ground beetle from Europe. Pterosticus melanarius, which is really big. And here's all the smaller predators that are small enough to feed on these root maggot flies. And what was happening is that the beetle banks were really, really good for this non-native beetle, and it was becoming really abundant, and these big beetles eat the little ones. So the reason it wasn't doing any benefit for pest control is the effective control agents were being eaten by another invasive predator that was accidentally being conserved. Okay, so just to kind of summarize a few of these key points from that brief overview of 5 years of poor Renee's life. First of all, the beetle banks were remarkably effective, and again, this is an on-farm data set, in increasing the numbers of predators. But because they were so attractive to this invasive predator that ate all the small predators, it actually didn't lead to any better pest control. And kind of the take-home from the center point of this whole session is that, this is the kind of thing you probably wouldn't ever figure out if you didn't do this work on farm, where you don't control what species are going to show up and what they're going to do. So the other part of this too, I guess I would say, is that we know that this is relevant to the real world because the work was done on an actual series of production farms. Okay, so the second question that I am to address today is what are some of the current collaborating farms and what are some of the research questions that you're doing? So Renee's work was, gosh, almost 14, 15 years ago now. But as I said, we still are doing all of our work now on real cooperating farms. So I want to kind of bring it up to some of the more modern day studies that we're doing. So what I'm going to do now is, this is again sort of a non-kind of traditional talk. So as I mentioned, I almost never go to the farms myself. But Joseph Taylor is here. He's one of my PhD students. And he's going to come up and just talk a few words about how, what we try to do to help the growers feel like they're really engaged in our work and we're not just kind of showing up on their farm, doing whatever and disappearing again, and kind of talk a little bit about why he was interested in doing an on-farm project. So for a lot of Joseph's work, he was working on farms all up and down the West Coast where he would live, out of a van or in a tent for 3 or 4 months at a time. So it's a pretty big effort for the students. So, Joseph.
Okay. Morning, everybody. Bill said, I'm Joseph Taylor. I'm a PhD student in this lab. And like he said, I was actually really, one of the reasons I came out to WSU to work with Bill is because he does on-farm research. And I could see some of the benefits of that. But first, talk about how we go about trying to keep growers excited and interested and willing to work with us year after year, and be accommodating to a bunch of scientists showing up on their farms and doing strange things. But the biggest thing, like with any relationship, is communication. So obviously, when you're doing research, and it's really those of you who've done research, it's easy to get kind of stuck in the writing the paper phase, and so between you doing the research and a paper actually coming out, there could be quite a bit amount of time, and then that paper isn't necessarily something that a grower would necessarily want or be interested in the finding, but necessarily be interested in trying to find and read. So what we were really tried to do, me and Olivia, was give the growers updates. So in our particular projects, we were looking at kind of biodiversity. So Olivia was focusing on birds, I was focusing on like insects. And so after we would survey a farm, we would make sure to compile the findings for that farm and then send it to the grower and kind of give them a little list of, these are the birds on your farm, these are like the insects we saw during the visual surveys on your plants. And if there was something particularly interesting like a bunch of mantids or something, because praying mantises are awesome, we'd let them know that and kind of email them back and forth and keep in touch, like how the project is going overall, how many other farms are going to, just making them, like Bill said, not just condensing them to another N in your data set, but making sure that you're actually keeping that human connection alive going forward. Another thing that's really helpful and great for allowing farmers to keep allowing you to go back to their farms is being flexible in your scheduling. It's not always possible. What we try to do is keep some wiggle room in our overall schedule so that if a grower what days work for you that we can come to your farm, and just being as convenient as possible for us to come and do science on their farm. Because certain growers, they have CSA days or that wouldn't work for us, that we would be in the way with our giant van of them doing what they need to do. So being flexible and willing to kind of maybe take a few hits on efficiency in order to keep heat from being inconvenient and making life difficult for the growers. That's like the number one rule is do not make lives difficult for the growers. Everything else you can work with, but you do not violate that. As to the rewards of field research, of on-farm research, for me personally, I did some work in undergrad that was all in like old fields in the middle of the woods that had no real impact on humans in general. Like, I thought it was interesting because I just like insects. I found them really fascinating. But the actual extension of like actually helping actual people out is really low in that kind of environment. So working in the real world, as Bill said, you can actually find out things that actually benefit real people. In a slightly more selfish note, I'm from the East Coast originally, so having to come out here and being forced to travel up and down the West Coast was kind of pretty cool. And so, but it was actually a really rewarding experience to meet a bunch of different kinds of people. Like, different kind of farming cultures that you go up Southern California all the way up to Northern Washington, and then realizing how similar people actually are even then with that huge spread of geography. So just getting to meet different people, get different takes on the world, knowing that your research is more directly applicable than it would be, and even though maybe like the cleanness of your data isn't as satisfying if you had a very sterile controlled experiment, but then again, as Bill just showed, your results from that kind of experiment are what might look nicer and make you feel better about it just because of the way it looks, but it's not necessarily very useful when you actually try to apply it to actual real-world research. So I kind of getting into rambling mode here, but so in short, things that we like to do to keep things keep growers in the loop and interested is keeping them in the loop, constant communication, and trying to be flexible in what we're asking of them, so that this is a partner relationship, not just us demanding things of the grower, because that's a really fast way to kind of sour the well on future research. And also, if you're considering doing on-farm research, I would highly encourage it, just because of one, you get to meet really awesome people with really good ideas, and also they get more than one occasion they'll give you, growers have their own insights of what happens on farms because they're there every day, kind of observing trends. And so a lot of research questions that our lab has kind of evolved forward with have come from direct observation from growers, and trying to incorporate into finding out exactly what's happening there, why do they see this. And being out on a farm, it's pretty fun. It's a lot more interesting than just being on either in a lab or on a very controlled lab university farm. So I don't know if Bill wants me to answer questions or.
No, I'll bring you back up at the end.
Okay.
I'm going to yank here. I'm going to go way over. Everyone's going to be angry.
All right. So thank you for your time. I'll pass things over to Bill.
I'll bring Joseph back up at the end. I'm going to try to time for questions. Okay, sorry, it's me again now. It's probably more fun to just listen to Joseph, but okay. So I just want to get back into some of the our modern day work. So towards this goal of trying to understand how useful biodiversity can be added to a farm, we've been looking at 3 different real-world farming systems. So the first, of course, is organic farming. We've been doing that work forever. The second one is these highly diverse mixed vegetable fruit farms. So oftentimes these are CSA or fresh market or farmers market supported farms that have an incredible amount of diversity on just a small piece of land. Diane Green's farm would be a great example. It's an acre or something like that, and she must have 20, 30 things growing at any given time. So there's with all that diversity come a lot of resources for natural enemies, of course. And the one that we've been working on probably the most lately is one of the more interesting types of farming systems that's becoming more common now, which is growers that have not just fruits and vegetables, oftentimes grains, but also have livestock integrated into their farming system as a source of fertility and also to provide meat products. So I like to say these farms are what we would now call an organic, mixed vegetable, integrated livestock, highly diversified farm. But 100 years ago, right, this would be called a farm, because essentially every farm in the world, and many still do, of course, look just like this. So there's some interesting rewards and challenges, though, that come with that farming system. And there's 3 systems components that we're most interested in. I think I'll get to just one of these today, but just to tell you what those are. We've been doing a series of projects looking at how the different manures and other fertility products that you add to the soil influence the ability of plants to not only defend themselves against herbivore attack. Of course, plants have sort of an immune system they use to defend themselves, but also their ability to attract natural enemies. So when plants are damaged, they often send out a help signal that attracts predators and parasitoids that can eat those pests. The second one is looking at the community ecology of food safety, and I'll talk about that in a minute. And then I'll briefly mention some of our work with wild birds that has been an extension of the food safety work. So again, what I did is I tried to pick out a couple farmers that I think the both our cooperators, and I believe will be here today. And you can see those folks here. Diane Green, as I've mentioned several times, has collaborated with us for years, although she's not pictured here. But across this West Coast work, we have probably at least 70 to 100. I've never really tried to tabulate it. Different cooperating growers that have helped with one or more of these projects through the years. And again, this is all on-farm research at a pretty massive scale, as I'll show you just real quickly with one example. So I'm going to talk about the food safety stuff. And again, of course, this is an example of a really bad type of biodiversity that we might not want. I know this is a topic that's on growers' minds a lot now because of the Food Safety Modernization Act and for other reasons. And it's a real particular challenge for small growers that don't have the ability to have a big sanitized cleaning facility and so on, like a huge producer might. It's just how to manage these food safety risks. So really, really bad things can happen if your produce becomes contaminated with E. coli or some of these other pathogens. People can get very sick or even die. And kind of beyond that, just from a pure practical matter, you know, if you're a farm that's doing a CSA and you make people sick, that's not going to be good for the bottom line of your farm. So it's something that nobody wants to ever get involved with. But, you know, how do you keep that from happening can be a real management and ecological challenge. So how do these contaminants get onto a farm? There's a whole wide variety of different places they can come from. Irrigation water, for example, people themselves that are out working in the crop. But there's two areas that we've been focused on that growers might have some ability to manage. Some of these can come from livestock, so you can make the decision to have livestock on your farm or not. But more difficult to manage is the case of wildlife bringing these things. So things like bird, deer, wild pigs can walk out into the field and poop, and when they do so, they bring the risk of contaminating your crops. So that's quite a difficult management challenge and really kind of goes at odds to the whole idea of what you want on your farm is to have more biodiversity. When you have hedgerows, natural plantings, ponds that are good for the beneficial aspects of biodiversity, you can attract wildlife that can potentially increase your food safety risk. So this has led to some of the dreaded so-called good agricultural practices or GAP regulations that many processors and other regulators are mandating for vegetable growers, where growers are being required to remove all natural habitats from their farms. So here's a highly diversified farm, biodiversity friendly. Here's one that's been modified by having the hedgerows bulldozed out. This is a deer fence here to keep any deer from coming in. And these pipes here are a trap. There's a snap trap in the middle here, and it's baited with grain. So if any rodents are on the farm, they're drawn in there and killed. So it's the idea that to minimize these food safety risks, you need to keep all wildlife off your farm and either kill or exclude them to make that happen. So big conflict with a lot of basic principles of organic farming. So I'm going to talk about some work that one of my PhD students is another case where a student brought a project into a lab, Matt Jones, who I'll show you in a second. And what Matt was thinking about is, you know, maybe this whole idea that you need to remove all biodiversity from the farm to improve food safety is completely misguided. Because, of course, there's a whole community of creatures out there that whenever they see poop, they say, delicious dinner time. So things like dung beetles are maybe particularly well known, I guess, and things that you can easily see. Of course, these beetles will come along, they'll ball up feces and bury them, and then have their larvae feed on them. And as part of that process, they would be removing them from contact with the vegetables, potentially. And then there's all kinds of creatures underneath the soil that also would use this resource, so things like bacteria, nematodes, fungi, that could antagonize those bacteria that are harmful and potentially kill them. So there's a natural defense potentially on a farm against food safety problems that could be harmed when you remove all natural habitats that support them. And so here's Matt. So John Reganold, a bunch of other people, a bunch of other professor people like me are on this project. But really, the truth is that Matt did all the work that I'm going to talk about. We just put our names on the papers and write the grants. So Matt had a pretty crazy project. So this is the scale of Matt's project. He had farms all the way from the Canadian border all the way down almost to the border with Mexico. He had farms under 3 farming systems, conventional vegetable farms, organic vegetables only, and then these highly integrated vegetable plus livestock farms. So you had 3 farming systems located all down the West Coast, and we did this over several years. What he did is, I always say, Matt had the most glamorous PhD project anyone's ever envisioned. He would fill a van with pig feces every spring. Then he would drive down to Southern California. He'd go out onto each one of these farms, and he'd put a little dollop of pig poop out on the farm field, and then he'd measure how quickly that was removed by the dung beetles and other creatures that would eat this stuff. I can quick show a video. The other thing he did then was also capture these beetles by using traps baited with pig feces. And I'll kind of just keep blabbing on. So this is a bag of pig poop here, and this is the trap. It's a little cup sunk in the ground. I don't know if you can see well, but that's a dung beetle right there. So it's like, delicious poop. It falls in the trap. Okay, so scary big figure. We couldn't get out of a figure by an ecologist without having one of these. So the big challenge that we have with these on-farm data is how do you make it into anything interpretable? And Matt was using an approach called structural equation models. So I won't take the time to go through this because that would be 45 minutes on its own right. And with the basic idea with these models is you put together a web of interactions that you think are important based on your hopefully expert knowledge, and then you use all the data you collect from a huge number of farms. You test the model with those data and see if your model actually is consistent with what you see in the field or not. Is kind of the basics of this. And these are very, very complicated, but it's two models. This is for the dung beetles, and this is for the soil microbes. And I'll just focus on kind of two take-home points from this. Both the communities of dung beetles above ground and the bacteria and other antagonists of these foodborne pathogens below ground were more abundant and diverse on the organic farms of either type, whether they were organic vegetables only or organic vegetables plus livestock. Above ground, there were bigger, more diverse communities of native dung beetles that are the most effective at removing Matt's little dollops of pig poop. And then below ground, there was more organic soil matter and more species of soil-dwelling bacteria on the organic than conventional farms. So both dung beetles and soil bacteria are more diverse and more plentiful on the organic than conventional farms. I'll just show you one result. This is survival of pathogenic E. coli in the soil. And on this axis, we're following the number of bacteria species. So Matt collected soil, we extracted DNA, and from those DNA, we can identify what bacteria species are there. And on farms that had more bacteria species in the soil, the E. coli had a more difficult time surviving. So these organic farms that have more soil biodiversity, it's more difficult for the foodborne pathogens to persist. And again, gosh, it's sort of horrifying to think. That was another 5 years of Matt's life. That was 3 slides right there. Because Matt just graduated. Anyway, the take-home from Matt, is that dung beetles and diverse soil microbes suppress these pathogenic E. coli and maybe other pathogens. Organic farming, these highly diversified farms attract more dung beetles and more species of soil bacteria that are more effective at killing off these potential foodborne pathogens. And again, highlighting the point that totally simplifying your farm and removing all biodiversity perhaps will distract wildlife or keep them from coming onto the farm, but you also might be killing the dung beetles and soil bacteria that are going to be your first line of defense. So it could be entirely counterproductive to do so. I'm going to see where I am here, because I was worried I might get long-winded. I fear that happened. I've got a couple minutes still. I'm still okay? Okay. So I'm just going to take just a few minutes to briefly summarize what's the very, very newest project that we're doing in the lab. This is another offshoot of this food safety work. Maybe I can do kind of a quick quiz. We got interested in what the views were of growers in particular of birds. So it's going to do a quick survey. I think we're about 50-50 growers and non-growers. So people that aren't growers, how many of you would like to have more wild birds in your yard or outside your house? People see if birds are good. Okay, of the growers, how many of the growers are here are hoping, I would really love to have a lot more birds on my farm? Okay. So we've got a few. How many growers would say, please, no birds? Yeah, I gave a talk a couple weeks ago to cherry growers in Wenatchee, and they were, I was going to told them I was going to give a little bit of talk about some of the benefits of birds, and they were very skeptical that there were any benefits of birds like cedar waxwings. So birds do a lot of things, and before I started doing all this on-farm work, I'd only thought of things like this bluebird here. Like, who wouldn't want this beautiful bluebird out there eating your pest insects? It's got to be all beneficial. But of course, birds do lots of other things. They don't just eat fruit, but they also can transmit these foodborne pathogens. They can bring parasites as a fowl mite that can get into your chickens and so on, even move around West Nile virus and some pretty nasty stuff. So, birds can do some beneficial and some harmful things on a farm. So with this A-Birds project, what we're trying to do is just get an idea of, on real working farms, what are the birds doing? So are they primarily eating pests and they're therefore beneficial? Or are they primarily transmitting foodborne pathogens and making food safety less certain? Again, this is another project, and this is the one that Joseph has been participating in, who talked to you a minute ago. Again, we're working at the scale of the U.S. West Coast. So we started doing more of these projects ever since I started just sitting in the office all the time and writing grants. These are great ideas in a grant proposal, but the students have to actually do it. Again, we have multiple farming systems, vegetables with integrated livestock. And what they're doing is they're going all down the West Coast. They're setting up mist nets, and they're capturing wild birds. Whenever you catch a wild bird in a mist net, it's pretty upset. When you pull it out of that, it will poop for you very readily. We collect those fecal samples, put them in alcohol, and bring them back to the lab. And then from those fecal samples now, through the wonder of modern sequencing approaches, we can extract DNA, and we can learn essentially everything that bird has eaten in the last 24 hours or so. We identify sometimes even a species, and we can also use the DNA to see if they have E. coli, Salmonella, or Campylobacter, some of these foodborne pathogens. So we're using birds that are on real working farms to learn about what their risks are posing to those same farms through these DNA approaches. So the DNA stuff has been another tool we can use now to do on-farm work in a real-world setting, but still have a lot of rigor to understand some of the mechanisms. And this is really early days for this project, but just a few data slides really quickly. So these are data for bird feces that were on broccoli leaves or near the plants. So this is the risk you're really worried about from a food safety perspective, is not whether or not a bird has ever been in your field, but whether it actually pooped on something that you're going to try to sell to someone. So here's just an example. So they're collecting these feces, extracting the DNA, and then seeing if the pathogens are there. And indeed, we did find a couple worrisome pathogens. So here are some of the data for Salmonella. And we were concerned, again, that the crop-only farms might have a lower risk than these crop plus livestock farms where we're actually potentially bringing the pathogens intentionally onto the farm. Okay, I'm showing this for two pathogens, Salmonella here, and these are only pathogenic strains of E. coli, so most E. coli that are in your body are not going to cause a problem. We're looking only for the ones that do make people sick. You can see that for both the Salmonella and the E. coli, we were finding them on these plants, the leaves of these plants. It looks, if you kind of squint, like maybe there's a higher risk in both cases on the crop plus livestock farms. What I want to stress is to bring your attention to what the y-axis is actually showing us. This is a really, really low probability of detecting these pathogens. So we collected about 1,000 of these bird feces at great effort. And I believe it was, Joseph can tell me this right, I think there were 4 total out of 1,000 that were positive for Salmonella, and it was maybe a dozen or so that had pathogenic E. coli. So now it gets into more of a sociological problem than an ecological one. So it is true that birds pose some level of risk, but what level of risk management or avoidance is really worthwhile to justify, this tiny, tiny needle in a giant big haystack that maybe there's going to actually be a bird depositing pathogens on your produce. Of course, these things will be washed and so on, so that they still could be removed at a later stage. Okay, last but not least, and I don't really have time left here to talk about this too much, we're also looking at what the birds are eating in their diets using DNA. And this is what we get. Oh my gosh, this is Joseph's project, is to figure out how to make sense of these data. But it's really, really complicated, the wonders and the limitations of modern DNA stuff. These are different species of birds. This is a savanna sparrow. This is white-crowned sparrow. These are all the different prey items that the birds are eating. The lines show when the thing was there or not. The brown things, I believe, are the insects, and the green things are the plants. And it's very, very, very complicated. But one thing I can point out is that we are seeing that birds, of course, eat not just pest insects, but they also eat a lot of predatory insects and spiders. So it's going to be an interesting project to figure out if their net impact is harmful or beneficial. Okay, so just conclusions. We did find these pathogens on the bird feces right in the foliage where you'd be the most concerned. Perhaps the livestock farms do have a slightly greater risk, but in all cases, the risk is incredibly low. Trying to see what I have there. And then to take you back to some of the mat stuff too, dung beetles and these pathogens would be, or these soil bacteria could be in place to even further lessen the risk on these highly diversified farms. Okay, what's convenient here is I realized, Joseph and I didn't talk at all about what he was going to say, but I realized that essentially everything that he said is already summarized here in the points that I separately came up with about why I think that this on-farm work is so rewarding and so interesting and why we've had to take over my research program altogether. And I'll just quickly summarize what Joseph said earlier. Obviously, these are actually results that are useful in the real world, so there's no artificiality or very little. And so hopefully the information we're generating can be used by growers to actually manage their farms without worrying if it's really relevant to what they're doing. And all these projects, to one level or another, have come from discussions with growers while we were doing something else. So part of why I was interested in food safety is because we had a big biocontrol project with a bunch of cooperating growers we brought together for an annual meeting to give us advice. I noticed when we didn't force them to talk about biocontrol, a lot of them were talking about food safety and what are we going to do to try to manage our food safety risk and meet these new regulations. And finally, some of these new statistical and molecular techniques let us do this work on a real-world scale and still understand some of the mechanisms that are interesting to us and help us publish in journals and all that kind of stuff. So, and at least it's easy for me to say, since I'm not the one doing all the work like the students are, but I think that there's less and less trade-off between having real scientific rigor and having real-world relevance because of some of these new tools. So you really can do high-impact science that gets into good journals and makes your career go well while still working on farms and doing things that actually benefit real people, as Joseph said. Okay, so how can growers get involved? Last but not least, I think that you will find if you're interested in doing this work, just contact a grower and say, hey, I'd like for you to come work on my farm. We're always going to be thrilled. And I'll just end with saying we're kind of weird, but not necessarily all that weird. So don't hesitate to approach us any chance that you get, and we'll be glad to work with you and try to help solve your problems. And so I don't know if I have time for any questions, but thanks for your attention. Okay. Oh, and I do want to point out all this work was funded by USDA. They keep getting closed down all the time, and I feel bad for all they do. None of this work at this scale would ever be possible without these larger grants that USDA has now through their organic program. So NIFA is really important, and I hope everyone continues to support their work and reach out to your legislators to let them know. And I was going to put this up. These are some online resources that the students have developed for birds, scouting for pest insects, and some information about dung beetles if you're interested. So, okay, now I'm really going to stop talking. Now there really is time for questions. Okay. Lynn. Yeah, a lot of this stuff is really, really scary. So Lynn's referring to, there have been a series of papers now at this point from all around the world where people have long-term data sets. Oftentimes it's in nature preserves, but the one in Germany I think is probably mostly farmland, I would assume, around there, where 50 or 60 years ago they'd put up traps and get X number of insects, and now they're capturing 90% less or some really, really scary number. Sometimes these are pretty large reserves. It really does kind of make sense that these kind of problems could be happening in the world because there's so many insecticides that are so widely available. Like I've heard some data that some of the most insecticide-contaminated water would be coming out of someplace like Seattle, where it's mostly homeowners that have a lawn care management company that comes, and the company is only going to get in trouble if somebody sees an insect, so they feel free to just nuke. A lot of farms are using a lot of insecticides, so it may well be at the point where insects in much of the world are really declining, and insects are the food for so many other things like birds. and ultimately benefit us in so many ways. So if that's really going on and those data continue to come in, that's obviously pretty troubling and is not a good sign for the world. It's kind of a dark note to end. Let's talk on another. Yeah.
So what would you see going forward that you think is the most important on-farm research that we should be doing?
Gosh, the question is, what's the most important on-farm research? I guess my answer would be the most important on-farm research is any on-farm research. I think that there's a fair number of us at WSU that do this. Probably WSU is better than most universities in the world for doing a lot of on-farm work, but a lot of people I think are intimidated as researchers to get into it just because people fear that it's going to take too long to get any papers and you're going to get fired and all the stuff I mentioned. So I think just kind of having a blueprint out there for how it can be done and still get the papers that you need for your career, but also be generating useful data. To get that word out there and get more people involved is kind of more of a need in my mind than any particular question. And plus, we've really never come up with the questions ourselves. We just listen to what the growers are worried about and kind of go from there. So, yeah. Yeah, that's a question I've never really gotten before. So why mostly west of the Cascades? So the project that I didn't have time to talk about, which is the soil health project, that has another whole block of farms, the cooperators that I showed, that are inland northwest as well. Some of the challenge, I guess, has been that this is so unique and probably uniquely different from what happens west of the Cascades, that within this block of projects that I talked in more detail about, we don't have transects on both sides. But a whole nother area of my research I didn't talk about at all is working on irrigated agriculture, conventional and organic in the Columbia Basin. And so we have tons and tons of on-farm work that we've done there. It's a whole different system, of course, as you know. It's often really big fields, and we've worked with potato growers and done a whole ton of projects that include on-farm components there. But I didn't even really mention that stuff here today. Should I stop? Okay, thanks again for your attention.